So today we are very glad to have Dr. Jeff Crater to teach data management, best practice for managed data in the lab and creating data management plan. Jeff, over to you.
Yep. Okay. Today I'm going to go over sort of management of data in the laboratory, and then also touch on data management plans. Just a brief history, sort of where I come from in regards to this, is that I've actually been involved in the development of a number of different data repositories, and have also worked on sort of curating data, and developing pipelines to curate data so that the data is understandable by people who, for example, then retrieve that data from the repository. Many different types of data, from imaging data to genomic data, time series data. So again, I've been involved in a number of different data repositories. So hopefully, what I cover today will be useful to you as well in the lab, and various practices that you can adopt, and also I just sort of general guidelines in terms of data management. So yesterday Mary Ann touched on sort of open science. Key part of that, as she mentioned, was open data. So the data that is that can be freely used, shared, and built upon, by anyone, anywhere, for any purpose. So, for example, there are many computational scientists out there, and if you have collected imaging data that you may not think is useful, it may be useful, for example, to a computational scientist. People who are looking at different computational methods for image manipulation or image filtering, tend to use lots of different types of data. So again, you may not see, even if you have data that is what is sometimes called negative data, or data that does not come to a conclusion. I'm publishing just data sets, which you can do nowadays, may be beneficial to someone else. And there are common problems with managing data over your lifetime in the lab. And so there's actually there's a very funny Twitter hashtag, overly honest methods. And so, here's a comment about, I can't send you the original data because I don't remember what my Excel file names mean anymore, right? So, 10 years ago, you generated, some data, named some, Excel files based on some of the data that you extracted from your experiment that you did the analysis on, but where are they? How do you find them? And so this is a common problem, in laboratory settings, and just in management of your own data files. And so yesterday, Mary Ann touched on, the different snafus with the video, but here, I have example of a spreadsheet that someone might have, might be generating from this. And so, if you're going back, or someone in the lab is going back, what in this case does age really mean? Can you tell? For example, if this is in humans, you might assume that age is in years, right? But if this is actually a mouse data set, it's probably not years, it's probably days, right? Gender, what was the encoding for gender in this case? What does a 1 or a 0 mean? Same thing with time and duration, right? So the documentation of your data is crucial in terms of making it understandable, and also making it understandable to others as well. And sharing is not just to make data public. So when we talk about data organization, how it can be used to make it more easy to share your data and understand your data, it's not just for external groups. So within your lab itself, it's you're collecting data, you need to make that data available to your PI, and so that he understands it. right? And so that that you can collaborate on that data. Another common situation in the lab is that there are multiple people collecting data for a study, right? So you need to make sure that everyone is on the same page, has the same protocol for collecting the data, naming the data, placing it into a shared space where whoever is then going to do the analysis, can do the analysis. As a graduate student in a lab, a lot of times, your first work might be using prior data collected in the lab, to familiarize yourself with, the studies, that have gone on in the lab, or to, learn some of the techniques that have been, developed and are used in the laboratory. So to make sure that that data is understandable for, new students coming in. And then the other, topic is, if you're collecting data across a number of different studies, at some point, you may be able to start pooling data from different studies to do analyses across data collected through those studies. So, I mean, there are a lot of reasons why, the concept of making your data understandable is important within a laboratory setting. So Marianne touched upon, the FAIR principles yesterday in terms of some of the key attributes for sharing. But, when one actually looks at sort of data itself, there are a lot of attributes that you have to start dealing with. And so there's, the data itself. So, how is that actually, formatted? There's the identifiers that Marianne talked about yesterday, the persistent identifiers. But then you get into, other types of documentation. So the use of, standards, open document formats, for storage of the data, and then also additional contextual information. So what is the larger scale metadata associated with the study and with the data that you are collecting. And a lot of times, some of this information may be collected in lab notebooks, on paper. So how do you encapsulate all this information within a data set, to make it understandable. In terms of metadata standards and ways to represent data, there's the fairsharing.org resource. And so this is a good resource for you to go to look for those types of standards. But what I really want to focus on today is sort of your responsibilities as a researcher, right, in terms of the FAIR principles. And I believe Marianne showed this yesterday, where there's responsibilities for different participants, in the FAIR ecosystem, right? You have the researchers who are, collecting the data, analyzing the data, the repositories that, store the data, and where researchers publish their data, and then the index aggregators. So these are the people that, make sure that the data is findable. And so as researchers, there are really, a number of topics, that we'll go through today. And so there's, good data management, the good documentation, providing rich metadata, making sure that you're preparing to share your data properly, and submitting it to a repository. And so good data management. So this is really, where it all starts. And this is really, the purpose here is to make sure that, any of the data that is collected, by you in the lab, is, high quality, well organized, and thoroughly documented, preserved and accessible, right? And so those are the key things, for data. And there are a number of practices that can be employed, and there are a number of motivations, right? So experiments done in the laboratory are complicated and can be organized in many different ways. So, there isn't, one way to organize, the data. If you're dealing with subjects or, for example, samples, you can be collecting multiple different types of data, multiple file types, multiple files, per subject or sample. And so again, this problem gets magnified by the types of data being collected, especially if you're collecting multiple types of data. You also have issues where if you're in a laboratory setting with different students, different postdocs, and there isn't a lab policy on data management, you're going to have different people collecting data and storing it in many different ways, and so that's again, this gets to the issue of understanding, being able to access data that you have not collected, making sure it's properly documented. And so that really gets to this difficulty in sharing data with and across large scale projects as well. So there are a lot of projects now, funded projects where multiple labs work together, either to collect data across a larger population, or to collect different types of data, with certain within certain populations. And so again, in these contexts, making sure that there's standardized protocols for the data and data management become very beneficial. And really, what this starts with is something that's very simple, right? It's making sure that you have some policy on file naming and folder hierarchies, right? And so I wanted to point out that many libraries, in this case the MIT libraries here, provide also guides in terms of data management. And so there are a number of different through this presentation, there are a number of different resources that I'll touch upon that give you the ability then to follow up and get more information. But the issue with file naming is important because that really lays the groundwork for being able to keep track of the data, documenting data. In this case, if you have standard file naming, folder hierarchies, you can easily point to information that's been collected within a study, based on what standards you come to in terms of file naming and folder hierarchy. And there's simple ways to do these types of hierarchies, and I'll get into an example in a couple slides. But the other important thing with having consistent naming and folder hierarchies is that as you collect data and you develop ways to analyze data, or QC your data, do quality control, or even develop tools to pre-process the data, to get it prepared for your analysis. You want to be able to do this in an automated fashion. And so having, consistent naming and folder hierarchies allows you to much more easily develop tools to run against the data that you're collecting, and apply them, to future studies as well, so that you, develop QC techniques for data that you're collecting. The next study, collecting the same type of data, you can apply the same QC tools to that data, because you have the same structure that you're running against. And so an example I want to get to is regarding imaging data. And there's a format that's called BIDS. So this is a data structure that has a lot of consistent naming of files, folder hierarchies, and what this affords is both that researchers have a very simple way to understand what the folder hierarchies are, so seen on the right here in the dataset file tree. But also, if you are to make any tools, or for example upload this type of data to a repository, it also provides for consistent ways then to develop interfaces or tools on top of that data. So on the left, you see sort of how a user interface for a repository can just pull the information from the data files that it expects to develop a user interface for that data. And again, once you have a standard format, there are other benefits as well. So, as people develop tools, and open science movement is not just about open data, but it's also about open tools, tools that are being made freely available. So if you have a consistent structure in a laboratory, for example, the tools developed by the different students, graduate students, postdocs, can be shared, can be easily used, across the different data sets. Development of validation tools is another important thing that I had mentioned. And a lot of these file formats or these standards, focus on the raw data, the minimally processed, but it's important to also keep in mind that these standards can and should be used for derived data as well. So again, if you're pre-processing the data, generating some analytic outputs from the data in terms of some of the processing scripts that are developed. And again, if you want to if you want to read up on this, again, this is a well laid out example with good documentation, so that's why I included it in these slides. But again, it's even though this is for imaging data, it's the same sort of scientific analysis workflow that everyone's involved in, right? You have acquisition, you're acquiring your data in a certain way, so you have parameters about the instruments you use and the like. That generates some raw data, which are then processed via some analyses or some pipelines that generate some derived data that end up in some publication. So again, these concepts for managing data, managing information about the data are fairly generalizable, right? And so BIDS itself, we talked about the directory structure, has a very prescriptive way of how you label directories within a data set to make it easy for you to find that. And then there's also information on how to label the actual individual files themselves, so that a human but also code can easily find and interact with that data. So for example, here you have so for example, if you're collecting data on subjects and either samples or sessions with that subject, the data type that's being collected, and then within that, so for example, if you have anatomical scans, you can then label them, including again the subject name in the file case, in this case within BIDS, because it makes it then easy to understand and always make sure that the file is associated with a particular subject. In terms of the higher level descriptions, this is where we get to then having other files that are available, to keep information about a study in the file system itself. And so, if you imagine that you collected data 3 years ago and you called the directory, study, 1 HD, you may not remember exactly what that study was, and you may have a lab notebook that has information on that that's buried somewhere in your bookshelf because it was a couple years ago. However, if you did contain a very simple manifest of information about that data set with on the file system itself, that makes it much more self-described, right? And you can have different types of information available. BIDS describes sort of a set of minimal information that's required for that. And you can store it in different ways. In this case, they're storing it sort of in JSON format, which is a simple data format that one can use, where you basically have field names, field names such as name, colon, and then the variable, comma, and the like. And you can have lists of items, where you have the square brackets. So there are a number of different ways to do this. You could also use spreadsheets, and I'll show an example of that a bit later on as well. For if you're dealing with subjects or participants, having a master file of the participants is very useful as well, because that can then help you run, for example, quality control checks across the data that you actually have, on the file system. So you're able to cross-check that, and you're also able then to store some of the information, for example, about the demographics or other data collected on the participants, that will allow you then to write, for example, analytics code, to either extract or do any type of analysis that you may need to do, with the data. And again, the participants files, these files can be spreadsheets. In this case, a tab delimited format, the TSV format. Again, having these data in standardly named places and using standard formats then allows, for example, a repository or any other tool that's developed to easily go into any study and render or access that information. Now, I mentioned that, BIDS was more imaging focused, but within the SPARC program that we're involved in, this is one of the NIH's Common Fund's large data collection programs. We've used the concepts that come from BID and have applied it to a much larger set of data, including genomics, time series data, other imaging modalities. And so, again, these concepts are very generalizable. And there is actually a white paper in bioRxiv, that describes sort of the SPARC data structure that's been developed, how it's applied, and tools that are being developed against that. So this is something that you can definitely, and I think I might have included this in the reading list. So this is a sort of a good white paper to get into as well, to look at that. And again, it's not BIDS exactly, but it looks very similar, right? So, in addition to subjects file, because, in SPARC, we're also dealing with samples. So for example, if you're dealing with genomic data or, doing, any, electrophysiology work, in vitro, any type of work where you're dealing with samples, those need to be described as well. So, again, that's one of the things that, was added in this case, because that is something, that's needed to describe the data set, but it's using the same generalized principles, right? So, just like, I described before, you had the data set and the subjects file, description files, we added a samples file. There's also additional files, in terms of, because this is a data collection, a program that supports large-scale data collection, there's also information researchers need to keep track on in terms of, their submissions, right? So again, using these generalized principles, all this, your structure for how you may want to store information about a data set can change. We also changed the folder structure a little bit, so that, that all the subjects and samples are held under a primary folder. Again, small changes, but it makes it easier, to navigate the data folders, so that they're not sort of overwhelmed with subject folders, because in some of the data collection studies, we have hundreds of subjects. And so this way, makes it a little more navigable for a human, when they're looking at it on their side. So again, utilizing these principles, you can come up with, your own, sort of structure, that will work, for yourself, in a larger scale, for the lab, potentially, and, has the important information that you need, right? And so, as you go through the data set, there's going to be, a number of required pieces of information. Again, this is the data itself and the core required metadata that you see on the top, but there are also a number of optional pieces that you can include as well. And so this is also important to define. So, having sort of a history log where you can add notes, for example, in a changes or a README file, is important. If you want to keep sort of ongoing information about, aspects of the data set. But then there are also optional areas that you can build on into the standard, right? So, for example, if there is specific code, that is, developed just for, this, maybe this data set in terms of analysis that's being done, that might be something that you can document in a code directory, that may even contain the code. If, for example, if the code is not stored in GitHub or some other code repository, derivative data, where do you store, the pre-processed data or the data that, the results of data that have been analyzed, does that go in a derivative folder, or, does that somehow get embedded, with the data itself? I mean, these are things that you need to work through in terms of, how you store your data. Supporting documentation, if you have a protocol in protocols.io, or you don't have a protocol in protocols.io and you want to document that somehow, where does that get stored? So again, there are a lot of things that one can work through in terms of deriving where, and how the data should be filed. And so, sort of to recap, really what you're trying to get to is a convention for organizing data into files and folders, and a set of descriptive files, that can fully describe the experiment that you can use, that helps you use your data and get back to the data, but also, helps you work with your data, as you're collecting it, so that, as you develop analysis code, you collect additional subjects or samples, right? You can run that analysis code automatically against those new subjects and samples, right? You don't have to do anything manually. And a key part of the management of the data is the documentation and rich metadata. So, we touched upon it briefly in terms of, some of the files that may contain some of that, but the data that you collect and the metadata that you associate with that data should really make this data self-describing, right? So in the subjects file, again, there's sort of things that you might consider to be required for, all the data that you're collecting. So, if you're looking at, subjects that are involved in, I mean, you're going to have a local subject ID, but then things such as, the age, the sex, what experimental group was the subject part of, species, strain, the RID for an organism, right? These are things that, you should always have, for for a subject, used in your experiment. But then you get to additional fields, right? So, what are some additional columns, that you may want to have? And so, that could be anything, so, this is just an example from Spark, where, body mass, was included for the organism. Same thing for samples. And for samples, you need to also sort of tie them to subjects, right? Because a sample has information as to, the organism. You might collect multiple samples from a single subject. And so in this case, you can have fields in your metadata, that basically link the sample to the subject ID, so that you can track these types of things. And again, then come the additional columns in terms of what is important for you. So, for example, one of them might be anatomical location. So if you're extracting samples, how do you describe where that sample was extracted from? And there are a number of different sort of minimal information standards. So I pointed you at fair sharing. And so you'll find there are many different types of minimal information standards that define sort of different metadata elements that one should collect. But again, this is dependent upon the study. So for example, if you're looking at the metadata related to, as I was talking about, the anatomical locations, right? You can look at, defining the anatomical location, defining, for example, a reference atlas was used, in terms of, that nomenclature, or extracting that information. Links to protocols. So this is another thing. So if you're, if you want to link a protocol to, a particular sample, you can do that as well by providing that information directly in the metadata file. And then also potentially, if there's, you want to, maintain information on, software versions that were used. So again, as software develops, analytic software develops, some versions of software end up having bugs, right? So, making sure you keep track of, what is the software version that you were using to do a specific analysis can become critical if it's found out that there is a certain type of bug that might have impacted your analysis. And so that's another thing to keep in mind. The data set description, again, this is something, where, there are, a number of sort of high level fields that you may want to get into. But you can also start tying in information, again, about, if an article, was published, using that data, you can include that then in the data set description file, so that, if someone in the lab comes back to this data, they can say, okay, this data was used, in this publication. So you can start linking, this information to make it more understandable. And so again, it can be a very simple spreadsheet, where you just sort of fill in the information. And the specifics, for example, within Spark that we have pulled into is that, since this is a large data collection effort, there's an issue of, for example, completeness of data. Right, so you may be collecting data in various batches, in terms of making data available, and so, in this case, we needed to add specific information. How does one, bring these data sets together? Again, if you're collecting data as part of a multi-center project, it's going to be the same issue, right? There's going to be a larger data set that your data is a part of, and that needs to be documented as well, to say that, this is part of a larger data set. And so if we go again into sort of the folders themselves, there is an optional sort of additional way to describe files. And so this is, doesn't always have to be done. But if you're generating, for example, different analytic outputs, and the file names can be maybe a little nebulous in terms of what analysis was done for a file, you may need to document that somehow. And so what you can do then is you can also include manifest files within directories. And a manifest file, again, can be something very simple. It just has the file name, the timestamp, and then maybe just a description. You can include a file type, depending on how, analysis software is written. Sometimes, again, the file types can be, can be nebulous as well. So again, this is something that can be included, when you're thinking about how you want to lay out your data. And, as you, work through, how, for example, the subjects and sessions, and data types, it's always good to sort of, work out, how it's going to support, the different types of data that you might be collecting, and working through, to make sure that, what you have for storing the data is will work for the data and the analysis that you're collecting. And so, there are different ways to, again, to do the layout. So for example, if you just have samples, and you don't really have subject data, you could have, just sample folders, but you still need to describe the subjects in some way at some point. So for data files, within the folders themselves, this is another area where you can develop certain specifications, again, to make it easier, for you to use the data and for you to actually work with the data yourself. So remember, these folder hierarchies and the file namings are not just, for yourself, they're for analysis applications, but they should also be understandable for you as you use the data. And so that's why there may be, depending on your lab and how data is collected, there may be different ways in which you want to name your files, so that that they're easily distinguished. And so there are different ways to do that. I mean, in some standards and hierarchies, you can recapitulate, for example, the subject and sample information in the file name, which makes it easier if you're doing, if you are just sort of in the shell doing sort of directory listings and finding data in that way, as a human would on their on their server, you would then be able to easily find the files just by directing your searches against the files themselves. So again, it really depends on sort of how you're working with your data, any of the other operating procedures for your lab, but how to make sure that those file names work for yourself and for any tools that you develop. And I did cover the data manifests, but again, one point here about the manifest is that you can provide a manifest at a higher level in the data hierarchy, right? So, if you had a lot of files, at the end in the data type directories, for example, in this case, where you might have, 4 different types of files, you can actually, in a more general way, define them at higher levels in the directory hierarchy. So if you want to say that, files with a certain extension or, a certain part of the name, you can do that, at higher levels in the directory. So that basically, that way you're basically saying that, any file that looks like this, right? So you can include, for example, in a manifest, you can include a file pattern, right? So if you're familiar with, working, in a terminal, if you do an ls to list your files, there are different ways to develop patterns, right, to describe a file. So like a star means match all. So if you did a star dot ext, right, that would mean that any file that has a name followed by the .ext extension, would match that pattern. And so you can describe, these files, sort of at a higher level as well. So again, there are different ways you can sort of mix and match, these principles. So having, the structure for the data is important, but then there's also, the importance of collecting data in a standardized way, right? So one of the critical things is, missing data, or if you're collecting data, if something went wrong in the collection of data and you have some files which are bad, right? But you but they shouldn't be used in the study. So you need to identify ways to notate that information, and that could be very simple in terms of, again, having metadata describing the samples. You can include something for a sample where you have some additional flags, for example, in the sample description file, or for example in a manifest file. If you have a manifest of related to the sessions or the data within that, right? And again, there are many different ways that you could do this, and so you would need to find a way that works for yourself and your laboratory. The important thing then is to during data collection using these standards is that once they're defined, that there's a way to enforce these standards. And one of the ways to do that is really through data quality, right? So if you have the ability to write scripts or tools to assess data quality, you can identify things such as missing data, right? Or additional data, right? And this is where having some flags, as we described above, for missing data or data that is something that describes that a certain set of data shouldn't be used, right? Because that might be the QC procedure might find additional data, but if that's already marked as not being usable, then the QC checks can still pass because it's only seeing the usable data. Sorry. The other part is checking for data with checking the data itself, right? So there may be certain QC checks that need to be done, to find anomalous values or to verify the data files have appropriate content, or if you're doing pre-processing and extracting the data for analysis, checking that data, the data that you've prepared for analysis, to make sure that the data is sound. And one way to do that, one way to do that is to actually do develop within your QC checks, some statistical summaries, to look for outliers. So again, applying this then to data collection, there are additional steps that may be needed. Now, depending on the type of data that you're collecting and what you need to do with that data. So remember, yesterday Marianne talked about, that data will need to be shared starting in 2023 with NIH's final sort of data sharing policies. Depending on the data you're working with, you may be required to submit the data to repositories that abide by certain data standards. And so this is additional information around how data should be described and how it should be described. And so again, that's something else that as you're developing data management practices for a laboratory, is to look at those data and information standards as well. And a key aspect of that is file formats, and to support the fair sharing of data. There are a couple aspects of file formats that you should be looking at, right? So that they're non-proprietary, that they're open and documented, commonly used, in the community. They have standard representations, and that there's no issues with the data being the data within the non-proprietary format is easily accessible, so that either it's unencrypted or encrypted using common tools. So for example, if you're using imaging data, there's TIFF and JPEG 2000, rather than GIF or JPEG. There's, instead of, for your data files, instead of Excel, especially the older, Excel formats, open formats that are based in ASCII, like CSV or TSV formats. And their ability is also to have data in multiple formats, right, in your data collection. So you could have an original format that may be proprietary, but you can convert those to a non-proprietary format and have those live sort of side by side, within the data itself. Again, to look and explore different data formats, you can again go to Fair Sharing, and they have a number of different file formats that they describe, everything from their genomic data standards, imaging data standards. So they have a number of different standards that they describe. And so again, this is a resource that you can use, to help work through some of that. Again, if you're looking at data collection all the way through sort of depositing in a repository, you can also start looking at the repositories to see what type of formats, they look at. And then, in addition to data formats, Fair Sharing also is a nice resource for various guidelines. So for example, what should be reported for in vivo experiments. So these are the ARRIVE guidelines that were developed, by a number of countries in the EU, in terms of reporting data for these types of experiments. There's also information about, for example, specific data types. So, the Miami standards for microarray data, giving you information as, what is expected with those data sets. Now, one key part of sort of defining your data is the use of controlled vocabularies, right? And so these are, controlled vocabularies really are, a way to standardize and organize the terms that you use to describe your data. And, when you're developing your metadata, using common terms from these vocabulary improves the ability to find the data, to aggregate the data, and is important, when you're collecting data, again, when multiple people are collecting data, that they're using the same terminologies. And, again, there are a number of different resources for this. You can, this is just an example. The libraries at University of Texas have an overview of controlled vocabularies. You'll hear the word ontologies as well. So controlled vocabularies, really, can be just as simple as, lists of terms, or lists of terms in a hierarchy. So, for example, if you're defining species, that you have mammals and you have rodents and you have rats and mice, so that is a type of hierarchy that you might have in a term list. Ontologies sort of build on top of controlled vocabularies with additional information. And so this is information, for example, if you have an ontology about cell types, that it might also have information that, that certain cell types are located in certain anatomical structures, right? So, really ontologies really start enriching term lists by including additional relationships, so they can relate, different types of terms to one another. And, there is the concept for, fair vocabularies as well. And, Marianne talked about this yesterday, where you have, that each concept, each term also has its own identifier, right? So that you're ensuring that you're making use of the same information. So whenever you document, for example, an anatomical location or a species, within some of the metadata files, you may want to include both the name and an identifier. So that that can be an unambiguous association. And when you use some of these terminologies in their native form. So these terminologies, a lot of come in different types of formats. And so one format is the resource description framework, RDF format. And, you can see that, in a term list, you might have, a name, synonyms, definition, its ID, that can also be machine readable. So in addition to the information, sort of the metadata in your data sets being machine readable, the information about any of the associated terms that you're using can also be machine readable. Again, this is getting to being able to automate various tools that you have in the lab. For example, making sure that if you're looking for all information that your lab has done around a certain anatomical location or a certain type of cell, right? If you're using the identifier, you can unambiguously know that all the data sets you pull back, that you point to with a certain identifier, do actually pertain to that anatomical region or cell type. And for the vocabulary, so if you have if you want to document the vocabularies, that you have the certain metadata about the vocabulary, so you can include things such as definitions, provenance, for example, if you're sort of describing sort of how your view of a term has evolved. So, for example, there are a number of terms that may be nebulous in terms of, for example, what substructures. So if you're looking at anatomical regions, what substructures a certain anatomical concept refers to. And so you can define that as well. That these terms have, the IDs. When you get these types of terminologies from some of the services that provide terms, they come in formal languages, but it can be as simple as just, again, a comma separated spreadsheet, that lists your terms. And so again, depending on what you need or what's required in terms of supporting the research that's going on, these things can be adjusted. To find terminologies, there are a number of resources out there. If you want to start, looking at, the terminologies and the identifiers, one of them is BioPortal. So this is at Stanford University. It's funded funded by NIH through a number of programs, and really they bring together a lot of different ontologies, terminologies developed in the biomedical field, and allows you to search across those. Within DKNet, we have Interlex, which is a terminology portal that tries to aggregate some of that information together. So, for example, if you're with diabetes, looking at the disease ontology, getting the identifiers there. So again, there are different resources that are available for you. Now, in terms of data, one of the last things I want to touch upon is simple things regarding sort of spreadsheets. So whenever you're collecting data, analyzing data, at some point, data is being transformed into a spreadsheet, either for, final statistical analysis or, some type of visualization. And so, there really are, a set of best practices, because there are things that, can cause spreadsheets to go a bit haywire, especially, dealing with gene names, sort of genetic sequence information and the like, because there are some things that the way spreadsheets format data that can end up being can cause some issues. And so there's actually, I think, an article last year that described actually some of those issues, but there are also other resources. So for example, within sort of this data carpentry, so data carpentry, has a lot of tutorials about working with data and developing tools around data. So again, the data carpentry sites are a good overall resource. In this case, there's a resource on spreadsheets. This one is developed through ecology, some ecology researchers. But again, these best practices are pretty generic. So you'll find different communities developing these tutorials around data, but a lot of them again are can be utilized in different fields. And so for example, here they two areas that we talked about already. So the formatting data tables, and then also quality control. But again, these are tools that you can use, and the data carpentry, as I mentioned, has a number of communities contributing, lessons, workshops, for building data literacy, in the fields, in the different fields. One part of developing good spreadsheets and good data files is your data dictionary, right? So the data dictionary is a collection, of the names, definition, and attributes about the data elements. And so if you are depositing data, for example, spreadsheets, a lot of repositories will require, a data dictionary. And a lot, it's the example at the very beginning, without a data dictionary, data can be, is not understandable. And so again, here's another resource. So UC Merced, has some information on developing data dictionary. So again, a lot of resources out there for you. One thing that you may become familiar with at some point then is that, when you're developing data dictionaries describing your data, that, you'll hear the term common data elements. And these common data elements are really elements that are defined within a community to describe, the same type of measure, the same type of data across studies. And so it's a standardized definition and relates to, a specific type of data, that is collected. And so, there will be common data elements, defining, for example, age, different types of demographics for human subjects. And, these are then, ensuring that that data, if it exists, in your data, that these common data elements describe the exact way in which that data was collected, and what the values mean. So from the original example, when we had gender at 1 and 0, right? The data element definition would define what 1 and 0 meant in that case. And there are different ways in which these are defined. So there are universal common data elements, right? And so if you look at the National Library of Medicine has resources that describe common data elements. There's a lot of work at NIH and many of the institutes developing common data elements, for what they study. And, so in addition to universal common data elements, so this might be high level demographic information, related to, for example, gender or age. There are also domain specific common data elements, that relate to specific topics or diseases. And then, there are going to be, certain required common data elements, and then also the concept of core data elements. For example, for certain types of genomic studies, there are going to be certain data elements that are going to need to be included. And there are a number of common data element collections and efforts. So again, if you go to NIH, there's the Phoenix Toolkit. There's PROMIS. There's an NIH toolbox that you can gain access to. There's work being done at NCATS, at NCI. So there are a number of different areas where these common data elements are being developed, and that you can reutilize in your research. The only issue is that you have to be aware that common data elements are not so common, in the sense that because common data elements describe specific ways in which a data element was collected, there may be multiple similar or very closely related data elements that may only differ, in terms of the types of values that field can contain, or the way that the data was calculated. And even when you look at age, for because you have a standard age metric, but then there's also age at diagnosis, age at death. So there are different types of ages that you may be dealing with. So just to make sure that you understand, if you start looking at the common data elements, that there are the same type. What you may think of being the same measure is not, even if it's collecting the same type of information. And in terms of working with your tabular data, there are now a number of toolkits that are being developed, that support working with tabular data, and working with tabular data in a way that is well-defined. So in this case, there's a frictionless data toolkit, which is supported, for example, within Python, within the Pandas toolkit. But they basically provide, a number of standards in terms of how to make your data packages, your tables self-describable, so that they can be easily used and brought into R, MATLAB, statistical packages, Python scripts, things of that nature. And it's an evolving toolkit, so it's a couple years old now, but it's really gaining momentum. So it's something that you can definitely take a look at. The next piece, so the final part of so the last 10 or so minutes, in terms of describing sort of the preparing to share your data, right? So in your lab, one of the key things is going to be ensuring data validity and sustainability, right? So this gets to storage and backup. So in addition to defining how your data should be stored, right, on the file system, you also need to look at, your data workflow and look at transformative steps in the workflow, right? So you may be collecting raw data. You may actually then recode some of the data, do some cleaning of the data. At what point, should that maybe be, maybe you don't want to save all the different steps of pre-processing. You may only want to store, the final step, or maybe there's an intermediary step that you want to snapshot the data at. So again, developing sort of the actual action plans around ensuring that your data doesn't get lost or get damaged is important. And then also, you may also want to have subsets of data for manuscripts. So if you have a much larger data set, let's say you're collecting a much larger data set, but you're only publishing, an initial subset of the data that's being collected. So maybe you collected certain genomic information, and you're doing analysis just on the genomic information, but you're collecting some additional longitudinal data from the subject. So you have an initial manuscript on the initial demographics and the genomics, but then you may also then have follow-up publication on the longitudinal data. You may want to subset some of that data so that you publish the data, because you'll be required, by certain journals to make that data available for that publication, so you'll need to subset that out of the larger data set. And so really this archival process is important. And nowadays, archival storage is pretty straightforward. There are many different options, everything from, Amazon, Google, Box, institutional facilities, and a lot of universities will, have licenses with cloud storage providers. So for example, in the UC system, we can archive into Google because that's something that is sort of site license within University of California. So check with your institution what options you have, because some of them may be low cost or no cost, for archiving your data. As Marianne pointed out yesterday, and I mentioned earlier, January 25th, 2023, is going to be a big change in terms of, the data, what's required, for your data. And so developing these data management and sharing plans that Marianne described is going to be very important in terms of ensuring that, you're prepared. So again, it's not just, because this is becoming more of a requirement, it's, not just, fulfilling the requirement that you need to have a data management and sharing plan, in your grant application, right? It's more important for you to have that worked out so that you understand what your requirements are and how you can meet those requirements with your data, because again, with the final rule, with more journals now requiring the sharing of data, to before your manuscript can get published, right? You need to know how you can, make sure that your data is shared appropriately. And so, there are tools to help you in developing data management plans. So, the DMP tool, has a number of different institutions that support this, but more importantly, there are public plans out there, that you can build upon, and they also have templates that you can use to help you get started. And so, the data management plan, in terms of what you need to provide, you need to provide a good description of, the types of data that you're collecting, the standards, that you're going to be used, the policies for accessing and sharing that data, plans for archiving the data, right? So where does this data go? Where is access going to be given, for that data? And so again, there are different ways to do that. There are other basic questions that you're going to need to answer in your data management plans. So, when will you make the data available, right? So this is usually upon publication and, a certain time, after conclusion of the data collection or of the study itself, right? So you're going to have to define, when you're going to make that data available, because, for example, not all data may end up being published, right? But depending on the NIH guidelines, because that data was collected, if that data was collected with NIH funding, even if you didn't publish on that data, you will still have to make it available, right? And so you need to describe that. How are you actually going to go about transforming the data for preservation? What documentation is going to be submitted alongside the data? Are there going to be any data sharing requirements? So, depending on the type of data it is, there may need to be some specific agreements in terms of access to the data. And so here, if you're sharing data with a known repository such as dbGaP or GEO at NCBI at NIH, they handle the data sharing agreements, right? So there are mechanisms within these repositories to handle researchers providing the appropriate, for example, IRB evidence that they have, have appropriate controls in place so that they can work with the data. And so again, that would be something where, if you're placing data in one of these repositories, those repositories will make sure to handle those issues. And so again, there's this DMP tool that's being made available. You can actually go in, and the nice thing is it keeps a dashboard, of different data management plans that you may be creating. So you can create data management plans for specific studies, and they have templates that you can build on, as I mentioned. And so it's a nice tool to keep that all organized. And it's a very simple site where you basically can create the new plans. You can select your university, your institution that you're working within, the funding agency, and then there are various templates it can provide. Now, since the new data sharing, the final rule came out earlier this year, there will be updates to many of the templates probably over the next year or so, as people start working with the new final rule. There are other resources. So NSF has actually a good resource in terms of describing some of these areas of a data management plan. So for example, describing, if you need to describe the data management in terms of roles and responsibilities, defining the key personnel who are involved in certain aspects of that. And again, a lot of this is, a lot of it may not be for public consumption, but they're, defining roles and responsibilities, for example, for data management is critical inside a lab, so that, people understand, who's, working on certain tasks. And if there's someone who is associated with quality control, what happens if, they're unavailable for a few weeks, who backs up, the quality control for a study, and especially if you're bringing in students as well who run some of these tasks, making sure that there's a way to provide information and manage what needs to be done for the different tasks. And so, with resource sharing plans, so this is the in addition to the data management plan, the resource sharing aspect of that. The sharing of that may depend on the types of data you collect. And here, again, we can talk about where DKNet can help, with the suggested data repository. So again, this is something that Marianne mentioned yesterday. But really, it's a way to, dig through the large number of repositories that are available and find out, the specific repositories, for example, for the types of data that you're collecting that are available. So I think that does finished exactly in 75 minutes. So that's I'm on time for once. And so, thank you, for listening, and hopefully, I've given you, some, outlines, best practices that you can apply, with your own data.